Okay, so pictured here is a cartoon of the modern nitrogen cycle. So nitrogen is an essential element for life. We need it for DNA, we need it for proteins. But even though our atmosphere is almost entirely composed of nitrogen, most organisms can't use it in this form, so nitrogen needs to be reduced or fixed. And this process is carried out by a small selection of microbes that basically provide all of the fixed nitrogen, nearly all of the naturally fixed nitrogen to the biosphere. So biological nitrogen fixation is also an exceedingly ancient metabolic process, and it originated with the evolution of the nitrogenase enzyme. So this is the only enzyme that's capable of fixing nitrogen, and it evolved somewhere between 2 and 3 or more billion years ago. And so really it's the activity of this nitrogen-fixing enzyme nitrogenase that has helped to sustain the biosphere over potentially nearly half or also perhaps the majority of Earth's history. All right, so pictured here on the left is the nitrogenase protein complex. Nitrogenase is also interesting because it's a metalloenzyme, and that means that it incorporates metal clusters in order to catalyze the reduction of nitrogen. So pictured here, this is the cofactor in this little insert image, and this particular cofactor is called the iron molybdenum cofactor, and it's called that because it incorporates one unique atom of molybdenum. That's the kind of teal-colored circle. But there are other forms of nitrogenase, and so the molybdenum 4 is by far the most common. It's also the most efficient. But there are also 2 other rarer forms, so they instead incorporate an atom of vanadium or an additional atom of iron in place of molybdenum. And so phylogenetic analyses have established that each of these 3 nitrogenase forms are genetically distinct, but they are evolutionarily related. And so then that presents the question, well, what was the metal usage of ancestral nitrogenases? And so the reason why this question is particularly interesting from an astrobiological perspective is it kind of requires that we probe this potential ancient interaction between marine geochemistry through time, as well as the evolution of nitrogen fixation and other metabolic processes. So these 3 metals that are incorporated in the nitrogenase cofactor are themselves redox sensitive. And so their abundances in the Earth's oceans through time have changed largely as a function of Earth's surface oxygenation. So for example, molybdenum is one example. Its abundance in the Earth's oceans was probably not very high until after the Great Oxidation Event. And so this observation then led to the hypothesis that perhaps ancestral forms of nitrogenase that originated prior to the Great Oxidation Event, maybe these were in fact molybdenum independent because there wasn't enough molybdenum to go around, basically. Still more recent phylogenetic analyses have instead suggested that the molybdenum form preceded the evolution of the vanadium and iron forms, and maybe this didn't happen until after the Great Oxidation Event, or somewhere close to it. So we wanted to further explore this history of nitrogenase metal usage, and we wanted to specifically be able to infer the metal dependence of reconstructed ancestral nitrogenase enzymes. And so we used a method called ancestral sequence reconstruction. So basically, this entails collecting a modern data set of, in this case, nitrogenase protein sequences, using that information to build a phylogenetic tree, and then using statistical methods to then infer an ancestral sequence that corresponds to some ancestral node of interest. And then with that sequence, you can analyze it for interesting features, and you can also use it to model ancestral protein structures. And so what we wanted to do is we wanted to use this method to specifically analyze the active site sequence and structural features that had previously been used to classify the metal dependence of modern nitrogenases and instead apply it to our inferred ancestors. So pictured here on the left, this is our nitrogenase phylogenetic tree. So this was built from a concatenated data set of molybdenum, vanadium, and iron nitrogenase protein sequences. Oops. So most of the tree is made up of molybdenum-dependent forms. I mentioned these are the most common. But up here on the top, this lineage, this is where the iron and vanadium nitrogenase forms cluster. And what's also interesting is they seem to branch after the divergence of this purple group here, MBMC. So this is another smaller group of molybdenum-dependent nitrogenases, and so this topology seems to be consistent with the idea that iron and vanadium forms evolved after the evolution of molybdenum usage. But at the base of this lineage are two other clades. So there's one in yellow, one in green. These are quote-unquote uncharacterized nitrogenases, and so they're called this because we don't have really any experimental evidence or any assessment of what metals that these nitrogenases incorporate. And so they're a little bit of a mystery. And so along this phylogenetic transect, we might expect some kind of historical change in metal usage. And so it's here where we specifically reconstructed and analyzed ancestral nitrogenase sequences in order to basically track this history of metal usage. Okay, so the first thing we wanted to look at was the active site sequence composition and see if we could find anything that appeared to correlate with metal binding. So on the bottom, these are pairwise comparisons of extant nitrogenase forms against each other. It's a heat map, so the lighter colors indicate that the active site sequence has lower identity, the darker colors indicate higher sequence identity. And so you can see that these comparisons do a decent job at differentiating the molybdenum forms from the vanadium or iron forms. And so we then bring that up to the top portion of this plot where we're comparing the ancestors, that's A through E, with the modern forms, and we find that most of the ancestors, B through E, seem to look a lot more like the molybdenum forms than the iron and vanadium forms. Perhaps with the exception of ancestor A at the top, which happens to be the direct ancestor of the iron and vanadium forms, and so its resemblance to those modern nitrogenase is quite reasonable. All right, so we also use these sequences to generate models of these ancestral proteins. So here they are, aligned and superimposed on one another. But we were specifically interested in calculating the volume of the active site pocket. And the reason we were interested, so that's what's pictured in pink right there, the reason we were interested again was that this parameter was previously used to classify metal dependence of modern nitrogenases. Again, we wanted to apply this to the ancestors. Okay, so in this rather complicated plot, these are pocket volumes both for ancestral nitrogenases and for extant nitrogenases. And I just want to draw your attention to the right side for the modern ones. And what was kind of surprising was that we see that the vanadium nitrogenases, as well as one major group of the molybdenum dependent ones, seem to occupy a very similar volume range. In fact, they're so similar and nearly indistinguishable that they're actually more similar to each other than are the two different groups of molybdenum nitrogenases. Also problematically is it seems like a lot of our ancestors also occupy the same range, and so kind of in contrast to the previous study, we don't seem to see any way to use this information to infer metal usage, particularly with regard to our ancestors. All right, so the last thing we did was we were interested in the distribution of proteins that today are involved with the synthesis of the molybdenum cofactor in nitrogenase. And so these two proteins, two of the various proteins that are involved in this pathway, NIF-E and NIF-N. So we mapped their distribution on taxa that are represented in our phylogeny. And what we found is that these two kind of strange clades, the yellow and green uncharacterized nitrogenases, they seem to have either a non-existent or incomplete set of NIF-E and NIF-N. And so by that information, you might infer that they are unable to use molybdenum. Perhaps parsimoniously, we might also infer that the oldest ancestors may similarly not have NIF-E and NIF-N, and so may be also molybdenum independent. Okay, so as a quick recap, it seems like our oldest ancestor active site sequence compositions resemble the molybdenum ones more than the vanadium and iron ones, but we have this seemingly contradictory observation that the basal uncharacterized nitrogenases lack the associated proteins for molybdenum dependence. So to solve this discrepancy, there's maybe two main options that we can go with. Perhaps these oldest ancestors did in fact use molybdenum, and these uncharacterized clades may have secondarily lost these cofactor proteins, or maybe they use a different pathway for molybdenum usage that we currently don't know anything about. The second option is that perhaps the oldest ancestors did not use molybdenum because they were not able to synthesize the molybdenum cofactor. They didn't have NIF-E, they didn't have NIF-N, and the sequence resemblances that we observe may not be entirely definitive, and that molybdenum usage really did evolve with NIF-E and NIF-N once molybdenum became available and not until after the divergence of these uncharacterized clades. So for a few reasons, we prefer the second scenario. The first thing is that we actually do have isotopic signatures of nitrogenase activity in the geologic record. So these were recently reported, and they date prior to our estimates for marine molybdenum availability, and they're also dated older than the molecular clock ages for NIFE and NIFN. Second, it's been previously proposed that nitrogenases prior to the evolution of NIFE and NIFN may still have been functional, so able to fix nitrogen. And this model kind of still leaves some room that it could be possible that early molybdenum usage may have been doable by some different, currently unknown pathway. So we can illustrate this model on our phylogeny. So first, potentially we started with a nitrogenase that had no associated NIFE and NIFN proteins, therefore was not molybdenum dependent, at least not in the typical way, and that this ancestral state is still exhibited by uncharacterized clades that still exist in the yellow. Then we have this stepwise addition of these cofactor proteins, perhaps starting with NIFE, and this is still reflected by the uncharacterized clades in green. And it wasn't until we have both NIFE and NIFN that today are harbored by the molybdenum-dependent forms in purple and also in blue that we had actual true molybdenum usage. And then finally, it wasn't until after the evolution of molybdenum usage that we get the diversification of the iron and vanadium forms. All right, so in conclusion, it seems like our ancestors, at least just looking at the sequences, seem to resemble molybdenum nitrogenases, but our phylogenetic analyses suggest that canonical molybdenum usage evolved with the cofactor synthesis mechanism NIFE and NIFN, and probably not until after the divergence of these uncharacterized clades. And finally, I just want to highlight that even though the work I presented here today is entirely computational, this method of ancestral sequence reconstruction also permits us to synthesize their encoding genes and quote-unquote resurrect these enzymes in the laboratory, and so we can continue to experimentally test these hypotheses. So please stay tuned for future work. Okay, so to just end here, I'd like to acknowledge our research group at the University of Arizona, particularly my PI, Batul Kajar. Please visit our website if you're interested in lots of other fun projects we're working on, and I'd like to acknowledge our funding sources, in particular the NASA Astrobiology postdoc program. So thank you very much.
We have time for a couple of questions.
Gene families are sometimes put into supergene families if you look deeper and you lower your homology threshold. And I'm wondering, have you looked at a possibly related gene family to the nitrogenases that you looked at? And if so, that would give you an outgroup for your tree.
Yes, so our tree is rooted with an outgroup. In this case, we used proteins they're called light-independent protochlorophyllide reductases. They're involved with the synthesis of bacterial chlorophyll, basically, and so they do share a distant homology. There's also other enzymes that seem to be fairly related. Those are called nitrogenase-like enzymes, and as of yet, I don't think we know exactly what they do, but they seem to be in methanogens.
Do they have any metal cofactors?
Yes, they do have metal cofactors. They do not look very much like the nitrogenase cofactor. I can't describe it to you off the top of my head, but yes, they are metalloenzymes.
Thank you.
Yeah. Would another possibility be that the ancestral nitrogenase sort of could use multiple cofactors, multiple enzymes, and that what you're seeing is sort of specializing and canalizing particular branches into being assembled in particular ways and maybe getting better as a result?
Yeah, so we had thought about this kind of interesting idea of like an ancestral generalist enzyme, and it's kind of cool to think about. It's a little bit difficult to reconcile with the modern processes of building that cofactor, which rely on a kind of very hierarchical structure of associated proteins that do the job basically. And so one potential option is that there was only a small subset of those proteins that directed perhaps just the core of the cofactor, deficient of molybdenum, maybe deficient of vanadium, but I'm not sure of any way that you could sort of get other metals transiently associating in the cofactor, but maybe by looking at these other uncharacterized nitrogenases, we can sort of expand our idea of the possibilities of how this nitrogenase could have functioned.
All right.
All right, if there are no other questions, let's thank again our speakers.